Dolly the sheep was not just any ordinary sheep. She was born on July 5th, 1996, at the Roslin Institute in Scotland. She was a scientific marvel, the first mammal cloned from an adult somatic cell. This means she was an exact genetic replica of another sheep. Dolly quickly captured the world's attention and became an icon of scientific progress. Her birth marked a pivotal moment in history, proving that scientists could create an entirely new individual from a single cell of an adult animal. Before Dolly, scientists had successfully cloned animals using embryonic cells. However, Dolly was different. She was cloned from a cell taken from the mammary gland of a 6-year-old Finn Dorset sheep. Dolly's arrival was met with a mixture of excitement and apprehension. The public was fascinated by the idea of cloning, with Dolly's name appearing in headlines worldwide. Despite the controversy, Dolly's birth was a watershed moment in scientific history. Dolly's creation was a triumph of scientific ingenuity, achieved through a process called somatic cell nuclear transfer. This technique involves removing the nucleus from an adult somatic cell and transferring it into an egg cell that has had its own nucleus removed. In Dolly's case, scientists at the Roslin Institute took a mammary gland cell from a 6-year-old Finn Dorset sheep. They then extracted the nucleus from this cell, which contained the sheep's DNA. Meanwhile, they took an unfertilized egg cell from a Scottish blackface sheep and removed its nucleus. The nucleus from the Finn Dorset sheep's mammary gland cell was then injected into the empty egg cell from the Scottish blackface sheep. This reconstructed egg cell was then given an electric shock to stimulate cell division and development. The resulting embryo was then implanted into the uterus of a surrogate mother, another Scottish blackface sheep. After a normal gestation period, the surrogate mother gave birth to Dolly, a healthy Finn Dorset lamb. Dolly lived her entire life at the Roslin Institute, where she was closely monitored by scientists and veterinarians. She enjoyed a comfortable and stimulating environment with plenty of space to roam and graze in the pastures surrounding the institute. Dolly was a curious and social animal, often seen interacting with the other sheep at the institute. Dolly's days were structured around her health and well-being. Scientists carefully monitored her diet, ensuring she received the necessary nutrients for a sheep of her age and breed. Regular health checkups were part of her routine, allowing the veterinary team to monitor her growth, development, and overall health. Despite being a clone, Dolly was able to live a relatively normal life. She mated naturally with a Welsh mountain ram and gave birth to 6 healthy lambs. Dolly's ability to reproduce naturally and give birth to healthy offspring was further evidence that she was a normal, healthy sheep, despite her unconventional beginning. Dolly's life at the Roslin Institute was a balance between scientific study and a normal sheep's existence. In her later years, Dolly developed osteoarthritis, a degenerative joint disease common in older sheep. This condition causes pain, stiffness, and reduced mobility, making it difficult for sheep to graze and move around comfortably. Dolly's osteoarthritis was first noticed when she was around 5 years old, relatively young for a sheep of her breed to develop this condition. Dolly's osteoarthritis was managed with pain medication and anti-inflammatory drugs, which helped to alleviate her discomfort and improve her mobility. Despite these treatments, Dolly's condition progressively worsened over time, leading to a decline in her overall health and quality of life. Some scientists speculate that it may have been related to her being a clone, as the process of somatic cell nuclear transfer can sometimes lead to genetic abnormalities. Despite the challenges posed by her osteoarthritis, Dolly continued to live a relatively comfortable life at the Roslin Institute. Dolly's story highlights the importance of providing proper care and management for animals with chronic conditions. On February 14, 2003, at the age of 6, Dolly was euthanized. This decision was made after a veterinarian diagnosed her with a progressive lung disease, and she began to show signs of severe distress. The news of Dolly's death was met with sadness from those who had cared for her and followed her story. Dolly's remains were preserved and are now on display at the National Museum of Scotland, a testament to her scientific significance and lasting impact on the world. Following Dolly's death, a post-mortem examination revealed she had a type of lung cancer called ovine pulmonary adenocarcinoma, or OPA. The discovery of this disease in Dolly raised concerns about whether her cloning process had made her more susceptible to illness. However, further investigation found no link between Dolly's cloning and her development of OPA. This finding helped to alleviate some of the concerns about the safety of cloning and reinforced the understanding that cloned animals like Dolly are susceptible to the same diseases as their non-cloned counterparts. Following Dolly's death, there was much speculation about whether her relatively early demise was linked to her being a clone. Some scientists hypothesized that the process of somatic cell nuclear transfer could lead to premature aging in cloned animals. This was based on the observation that Dolly's DNA showed signs of shortened telomeres, which are protective caps on the ends of chromosomes that shorten with age. However, further research has shown that telomere length in cloned animals can vary significantly and is not always a reliable indicator of aging or lifespan. Studies on other cloned animals have not shown a consistent pattern of premature aging or shortened lifespans. In fact, many cloned animals have lived long and healthy lives, suggesting that Dolly's case may have been an anomaly rather than a reflection of inherent risks associated with cloning. While Dolly's case raised important questions about the long-term health of cloned animals, subsequent research has largely dispelled the myth that cloning inevitably leads to premature aging or other health problems. Like any scientific breakthrough, the development of cloning technology requires ongoing research and careful observation to fully understand its potential benefits and risks. Dolly's birth marked a watershed moment in the field of biotechnology, proving that it was possible to clone mammals from adult cells. This breakthrough ignited a flurry of research into cloning other species, leading to the successful cloning of a variety of mammals, including cows, pigs, goats, mice, and monkeys. Each successful cloning attempt brought with it new insights into the complexities of cellular development and differentiation. The ability to clone mammals has significant implications for various fields, including medicine, agriculture, and conservation. In medicine, cloning technology holds promise for organ transplantation, gene therapy, and the development of new treatments for diseases. In agriculture, cloning could be used to create copies of animals with desirable traits, such as high milk production, disease resistance, or superior meat quality. However, the use of cloning in agriculture raises ethical concerns about animal welfare and the potential for reducing genetic diversity in livestock populations. Conservation efforts could also benefit from cloning technology, particularly in the preservation of endangered species. While cloning alone cannot save endangered species, it could play a role in preserving genetic diversity and providing a safety net against extinction. Dolly the sheep's legacy extends far beyond the realm of science. She became a global icon, capturing the imagination of people worldwide and sparking conversations about the ethics and implications of cloning. Her name became synonymous with scientific progress, often used as shorthand for the potential of biotechnology to reshape the world. Dolly's image graced the covers of countless magazines and newspapers, and her story was featured in documentaries, television shows, and even a Hollywood movie. Dolly's impact on science is undeniable. Her birth challenged long-held scientific dogmas about the nature of cellular differentiation and opened up new avenues for research in developmental biology, genetics, and regenerative medicine. Dolly's legacy continues to inspire scientists today as they explore the potential of cloning and stem cell research for medical breakthroughs and the preservation of endangered species. Dolly's story also serves as a reminder of the importance of ethical considerations in scientific research. These conversations continue today as scientists grapple with the ethical implications of new biotechnologies such as CRISPR-Cas9 gene editing. In December 2023, a poignant tribute to Dolly's legacy unfolded at the National Museum of Scotland, where her preserved remains have been on display since 2003. Dolly's fleece, carefully sheared during her lifetime at the Roslin Institute, was unveiled as part of the museum's collection, offering a tangible connection to the iconic sheep. The fleece, a soft and woolly reminder of Dolly's physical presence, is now displayed alongside educational materials that delve into her scientific significance, the cloning process, and the ethical debates surrounding her creation. This addition to the museum's collection provides visitors with a unique opportunity to learn about Dolly's life and legacy, fostering a deeper understanding of the science behind cloning and its impact on society. The fleece serves as a tangible link to the past, reminding us of the groundbreaking scientific achievement that Dolly represented and the ongoing impact of her legacy on the world. Dolly's story, from her unconventional birth to the final tribute of her fleece on display, continues to captivate and inspire.